एनाटॉमी ऑफ फ्लावरिंग प्लांट्स 6 नंबर का हमारा चैप्टर है. You can very easily see the structural similarities and variations in the external morphology of the larger living organism, both plants and animals. Similarly, if we were to study the internal structure, one also finds several similarities as well as differences. This chapter introduces you to the internal structure and functional organization of higher plants. Study of internal structure of plants is called anatomy. Plants have cells are organized into organs. Different organs in a plant show differences in their internal structure within angiosperms. The monocots and dicots are also seen to be anatomically different. The internal structures also show adaptations to diverse environments. The tissue system. We are discussing types of tissues based on the types of cells present. Let us now consider how tissues vary depending on their location in the plant body. Their structure and function would also be dependent on location. On the basis of their structure and location, there are three types of tissue systems. These are the epidermal tissue systems, the ground or fundamental tissue system, and the vascular tissue system or conducting tissue system. Epidermal tissue system. The epidermal tissue systems forms the outermost covering of the whole plant body and comprises epidermal cells, stomata, and the epidermal appendages, the trichomes and hairs. The epidermis is the outermost layer of primary plant body. It is made up of elongated, compactly arranged cells which form a continuous layer. Epidermis is usually single layered. Epidermal cells are parenchymatous with a small amount of cytoplasm lining the cell wall and a large vacuole. The outside of the epidermis is often covered with a waxy thick layer called the cuticle, which prevents the loss of water. Cuticle is absent in roots. Stomata are structures present in the epidermis of leaves. Stomata regulates the process of transpiration and gaseous exchange. Each stoma is composed of two bean-shaped cells known as guard cells, which enclose stomatal pore. In grasses, the guard cells are dumbbell-shaped. The outer walls of guard cells waxy away from the pore and thin, and the inner walls towards the stomatal pore and highly thickened. The guard cells possess chloroplasts and regulate the opening and closing of stomata. Sometimes a few epidermal cells in the vicinity of the guard cells become specialized in their shape and size and are known as subsidiary cells. The stomatal aperture, guard cells, and the surrounding subsidiary cells are together called stomatal apparatus, which is located in figure 6.1. Stomata with bean-shaped guard cells, stomata with dumbbell-shaped guard cells, bean-shaped guard cells, and dumbbell-shaped guard cells. The cells of epidermis bear a number of hairs. The root hairs are unicellular elongations of the epidermal cells and help absorb water and the minerals from the soil. On the stem, the epidermal hairs are called the trichomes. The trichomes in the shoot system are usually multicellular. They may be branched are unbranched and soft and stiff. They may even be secretory. The trichomes help in preventing water loss due to transpiration. The guard tissue system. The tissues except epidermis and vascular bundles constitute the guard tissue. It consists of simple tissues such as parenchyma, collenchyma, and sclerenchyma. Parenchymatous tissue cells are usually present in cortex, pericycle, pith, and medullary rays. In the primary stems and roots. In leaves, the ground tissue consists of thin-walled chloroplasts containing cells and is called a mesophyll. The vascular tissue system. The vascular system consists of complex tissue, the phloem and the xylem. The xylem and the phloem together constitute vascular bundles, which is located in figure 6.2. In dicotyledonous stems, cambium is present between phloem and xylem. Such vascular bundles, because of the presence of cambium, possess the ability to form secondary xylem and phloem tissues and hence are called open vascular bundles. In the monocotyledonous, vascular bundles have no cambium present in them. Hence, since they do not form secondary tissues, they are referred to as closed vein xylem and phloem within a vascular bundle are arranged in an alternate manner along the different radii. The arrangement is called a radial, such as in roots. In conjoined type of vascular bundles, the xylem and the phloem are jointly situated along the same radius of vascular bundles. Such vascular bundles are common in stems and leaves. The conjoint vascular bundles usually have the phloem located only on the outer side of xylem. Anatomy of dicotyledonous and monocotyledonous plants. For a better understanding of tissues, organization of roots, stems, and leaves, it is convenient to study the transverse sections of the major zones of these organs. Dicotyledonous root. Look at figure 6.3a. It shows the transverse section of the sunflower root. The internal tissue organization is as follows. The outermost layer is epiblema. Many of the cells of epiblema protrude in the form of unicellular root hairs. The cortex consists of several layers of thin-walled parenchyma cells. With intercellular spaces, the innermost layer of the cortex is called the endodermis. It comprises a single layer of barrel-shaped cells without any intercellular spaces. The tangential as well as radial walls of the endodermal cells have a deposition of water impermeable waxy material suberin in the form of Casparian strips. Next to endodermis lies a few layers of thick walled parenchymatous cells referred to as pericycle. Initiation of lateral referred to as pericycle. Initiation of lateral roots and vascular cambium during the secondary growth takes place in these cells. The pith is small and inconspicuous between the xylem and phloem are called conjunctive tissue. There are usually 2 to 4 xylem and phloem patches. Lateral cambium ring develops between the xylem and phloem. All tissues on the inner side of the endodermis, such as pericycle, the vascular bundles, and pith constitute the stele. Monocotyledonous roots. The anatomy of the monocot root is similar to the dicot root in many respects. Figure 6.3b. It has epidermis, cortex, endodermis, pericycle, vascular bundles, and pith as compared to the dicot root, which have fewer xylem bundles. There are usually than six more than 6 polyarch xylem bundles in the monocot root. Pith is large and well developed. Monocotyledonous roots do not undergo any secondary growth. Dicotyledonous stem. In the transverse section of the typical young dicotyledonous stem have shows that the epidermis and the outermost protective layer of the stem, which is located in figure 6.4a, dicot, covered with a thin layer of cuticle. It may be trichomes and a few stomata. The cells arranged in multiple layers between epidermis and pericycle constitute the cortex. It consists of three subzones. The outer hypodermis consists of few layers of collenchymatous cells just below the epidermis, which provide mechanical strength to the young stem. Cortical layers below hypodermis consist of the rounded, thin-walled parenchymatous cells with conspicuous intercellular spaces. The innermost layer of the cortex cells with the conspicuous intercellular spaces. The innermost layer of the cortex is called the endodermis. The cells of the endodermis are rich in starch grains, and the layer is also referred to as the starch sheath. Pericycle is figure 6.4. Stem of dicot and stem of monocot. Present on the inner side of the endodermis and above the phloem in the form of semilunar patches of sclerenchyma. In between the vascular bundles, there are few layers of radially placed parenchymatous cells which constitute medullary ridge. A large number of vascular bundles are arranged in the ring. The ring arrangement of vascular bundles is a characteristic of dicot stem. Each vascular bundle is conjoined, open, and with enlarged protoxylem. A large number of rounded parenchymatous cells with large intercellular spaces, which occupy the central portion of stem, constitute the pith. Monocotyledonous stem. Monocot stem has a sclerenchymatous hypodermis, a large number of scattered vascular bundles, each surrounded by a sclerenchymatous bundle sheath, and a large conspicuous parenchymatous ground tissue, which is located in figure 6.4b. Vascular bundles are conjoint and closed. Peripheral vascular bundles are generally smaller than the centrally located ones. The phloem parenchyma is absent and the water containing cavities are present within the vascular bundles. Dorsiventral dicotyledonous leaf. The vertical section of the dorsiventral leaf through the lamina shows three main parts, namely epidermis, mesophyll, and vascular system, and the endoepidermis, which covers both upper surface adaxial epidermis and the lower surface abaxial. of the leaf has a conspicuous cuticle. The abaxial epidermis generally bears more stomata than the adaxial epidermis. The latter may even lack stomata. The tissue system, the tissue between the upper and the lower epidermis is called the mesophyll. Mesophyll, which possess chloroplast and carry out photosynthesis, is made up of parenchyma. It has two types of cells, the palisade parenchyma and the spongy parenchyma. The adaxially placed palisade parenchyma is made up of elongated cells which are arranged vertically and parallel to each other. The oval and round and loosely arranged spongy parenchyma is situated below the palisade cells and extends to the lower epidermis. There are numerous large spaces and air cavities between these cells. Vascular system include vascular bundles which can be seen in the veins and the midrib. The size of vascular bundles are dependent on the size of veins. The veins vary in thickness. In the reticulate venation of the dicot leaves, the vascular bundles are surrounded by a layer of thick-walled bundle sheath cells. This is a look at which is located in figure 6.5a and find the position of xylem and phloem. Xylem in the vascular bundle. Isobilateral monocotyledonous leaf anatomy. The anatomy of isobilateral leaf is similar to that of the dorsiventral leaf in many ways. It shows the following characteristic differences. In an isobilateral leaf, the stomata are present on both the surfaces of the epidermis, and the mesophyll is not differentiated into palisade and spongy parenchyma, which is located in figure 6.5b. The grasses contain certain adaxial epidermal cells along the veins, modify themselves into large empty colorless cells. These are called bulliform cells. When the bulliform cells in the leaves have absorbed water and are turgid, the leaf surface is exposed. When they are flaccid to water, the parallel venation in monocot leaves is reflected in the near similar sizes of vascular bundles except in main veins as seen in vertical sections of the leaves. Summary: anatomically, the plant is made of different kinds of tissues. The plant tissues are broadly classified into meristematic, apical, lateral, and intercalary, and permanent minerals and photosynthetes. The mechanical support are the main functions of tissues. There are three types of tissue systems: epidermal, ground, and vascular tissue system. The epidermal tissue systems are made of epidermal cells, stomata and the epidermal appendages. The ground tissue systems forms the main bulk tissue system is formed by the xylem and phloem on the basis of presence of cambium location of xylem and phloem. The vascular bundles of different types. The vascular bundles form the conducting tissue and the translocate water, minerals, and food material. Monocotyledonous and dicotyledonous plants show marked variation in their internal structures. They differ in type, number, and location and vascular bundles. The secondary growth occurs in most of the